Welcome to the Deep Dive. Today we're tackling a subject that's absolutely vital to understanding our health: the diseases of the immune system. Think of your immune system as this incredibly intricate, really powerful defense force. It's constantly working to shield you from, well, everything from tiny bacteria and viruses to even those rogue cells within your own body that could potentially turn into cancer. But like any complex system, it can sometimes malfunction. It might underreact, leaving you vulnerable, or maybe overreact and actually cause harm itself.
Exactly. And to really truly master the complexities of these diseases, we absolutely need to build a strong foundation in how the immune system is supposed to function normally. We'll be drawing on a pretty comprehensive pathology textbook as our source today. The aim here is really to achieve that kind of deep understanding the level an elite professional in the field would possess. Our goal is to equip you with that level of knowledge.
Okay, let's unpack this now. We're not just going to skim the surface here. This is a proper deep dive
Okay.
into the whole spectrum. We'll start with the normal immune response. It's our baseline, what should happen, and then really explore what occurs when things go wrong, leading to immunodeficiency, where the system is too weak, or hypersensitivity, where it gets, well, overzealous.
Right. And as we explore, you'll see that understanding the individual components, the various cells, the tissues involved, even the microscopic molecular receptors and how they all interact in a healthy immune response is just fundamental to grasping why and how these diseases develop. We'll be systematically examining the two major branches: innate and adaptive immunity, all the diverse players within each branch, and the absolutely crucial role of the major histocompatibility complex, or MHC.
Okay, so our mission today is a deep immersion into these core immunological principles. By the end, you should have a really robust understanding of how the immune system works, so that when we discuss its various disorders, it all just clicks together, like it would for a seasoned professional, ready to get into the weeds.
Absolutely. Let's begin with the body's immediate, ever-ready defense system: innate immunity.
Innate immunity, that's like the security guards always on duty, yeah. The body's first responders, ready to act instantly against any kind of perceived threat.
Precisely. It's the body's initial line of defense, and it's comprised of several key elements. Think first of the physical barriers, like your skin, the linings of your respiratory and digestive tracts. Those are the first walls. Then you have various types of cells: phagocytes, which are like little Pac-Men engulfing foreign material; innate lymphoid cells, or ILCs; natural killer cells, NK cells; and also crucial plasma proteins, like the complement system.
And within this innate system, there are these fascinating cellular receptors. They sound almost like something out of science fiction. Toll-like receptors, NOD-like receptors, what exactly are they sensing?
Well, what's truly remarkable is how these receptors let the innate immune system immediately recognize danger signals. Toll-like receptors, TLRs, they act like sentinels. You find them either on the cell surface or inside, in compartments called endosomes. They're designed to spot specific molecular patterns that are common to lots of different microbes, things like bits of bacterial cell walls, or, say, the genetic material of viruses. And importantly, they can also recognize molecules released by our own cells when they get damaged or stressed.
Okay.
So when a TLR detects one of these signals and gets activated, it kicks off a signaling cascade inside the cell. This often leads to producing antiviral proteins called type I interferons, really critical for that early defense against viruses. And as you said, their importance is clear. Loss of function mutations in TLRs, or the pathways they trigger, can cause serious immunodeficiency syndromes. It really highlights how vital these early warning systems are.
So TLRs are looking for these broad, sort of generic enemy signatures. What about NOD-like receptors, NLRs? They sound like they might be looking for something different, maybe more specific.
Not necessarily more specific to the type of microbe, but they operate in a different location inside the cell, in the cytoplasm. NLRs also recognize a diverse array of danger signals. These often include things released from cells that are dying or damaged, like uric acid, ATP, even the loss of potassium ions from inside a cell. And, of course, certain microbial products that have managed to get inside. Exactly how they sense such a wide variety is still, a very active area of research.
Right.
Now, here's where it gets really interesting and connects to inflammation. Some of these NLRs can activate a large multi-protein complex called the inflammasome.
Inflammasome, that sounds like a pretty serious piece of cellular machinery. What does it actually do once it's activated?
Its main job is to activate a specific enzyme, caspase-1. Think of caspase-1 like molecular scissors. It cleaves an inactive precursor form of the signaling molecule interleukin-1, or IL-1, into its active form. Active IL-1 is a major driver of inflammation. It helps recruit other immune cells to the site of injury or infection, and it's also a big contributor to fever. Now, what's crucial to understand here, especially to distinguish it from autoimmunity later, is that gain-of-function mutations in NLRs or loss-of-function mutations in the proteins that regulate the inflammasome can cause these conditions called autoinflammatory syndrome.
Okay, autoinflammatory, not autoimmune.
Exactly. They cause episodes of intense, seemingly unprovoked inflammation, but they don't primarily involve the adaptive immune system attacking self-antigens like autoimmune diseases do.
Got it. So innate immunity is this rapid, general, always-on first response. Then we have adaptive immunity, which is more like a specialized, highly trained force that develops over time. What are the really fundamental differences between them?
The defining features of adaptive immunity are its incredible specificity and diversity in recognizing, well, a virtually limitless range of antigens, any foreign molecule the body might meet. And it has this unique ability to remember prior encounters. This immunological memory allows for a much faster, dramatically stronger response if you meet the same threat again. That's the whole basis of vaccination and long-lasting immunity.
And the key players here are lymphocytes, T cells, and B cells. They might look kind of similar under a microscope, but their jobs and how they spot threats are vastly different, aren't they?
Absolutely. They come from common precursors, but T and B lymphocytes are incredibly diverse and specialized. T lymphocytes, T cells, mature in the thymus. They express specialized surface receptors called T cell receptors or TCRs. Now, TCRs have a very specific way of seeing antigens. They only recognize small peptide fragments from proteins. And critically, these peptides must be displayed on the surface of other cells by these molecules called the major histocompatibility complex, MHC.
MHC.
Think of MHC like the display cases presenting these peptides to the T cells.
Right, MHC sounds absolutely central. We'll definitely need to dig into MHC in detail. But first, more on these T cell receptors and the different kinds of T cells.
Okay, so each TCR is a complex structure itself. It's made of two protein chains, alpha and beta, forming a heterodimer. That's the part that actually binds the peptide MHC complex. Importantly, this TCR is linked non-covalently to a cluster of other invariant proteins, meaning they're the same in all T cells, called the CD3 complex and a zeta chain dimer. These CD3 and zeta proteins don't bind antigen. Their job is to transmit signals into the T cell once the TCR recognizes its specific antigen on an MHC molecule.
Okay, the signaling part.
Right. Now, there are two major functionally distinct subsets of T cells we need to focus on: CD4 positive, CD4 plus T cells, and CD8 positive, CD8 plus T cells. These are mutually exclusive. A mature T cell is either CD4 or CD8, not both. In a healthy person, CD4 plus T cells are about, say, 60% of mature T cells. Their main role is as helper cells. They secrete signaling molecules called cytokines, which help activate and direct other immune cells, like macrophages and B cells, to fight infections. CD4 plus T cells are specialized to recognize peptide antigens presented by class-high MHC molecules. They use their CD4 coreceptor protein to specifically interact with these class-high MHCs, which is crucial for starting the activation signals. That's why we call them class-high MHC restricted.
And CD8 plus T cells. Their main job is to directly kill infected cells, if I remember that correctly.
Precisely. CD8 plus T cells, usually about 30% of mature T cells, function as cytotoxic T lymphocytes, or CTLs. Their critical role is to recognize and directly destroy host cells infected with intracellular microbes, like viruses, or even cells that have become cancerous and are displaying abnormal proteins. CD8 plus T cells recognize antigen peptides presented by class I MHC molecules. They also have the CD8 coreceptor protein, which binds specifically to class I MHC and is essential for getting the signals needed for activation and killing.
So they are class I MHC restricted.
Exactly. This restriction makes biological sense. CD8 plus cells check the inside of all our cells for trouble, while CD4 plus cells orchestrate responses to threats usually found outside cells and processed by specialized antigen-presenting cells. And besides CD4 and CD8, other proteins like CD28 and integrins are important too. CD28 receives extra activation signals co-stimulation from APCs, preventing inappropriate activation. Integrins are adhesion molecules, helping T cells stick to APCs for proper recognition.
Okay, so T cells need these peptide MHC complexes. What about B lymphocytes? Their receptors work differently, right?
Yes, B lymphocytes, B cells, have a fundamentally different way of seeing antigens. They are unique, the only cells producing antibodies or immunoglobulins. These are the key players in humoral immunity mediated by soluble factors in the blood. B cells develop in the bone marrow. Mature, naive B cells circulate and hang out in peripheral lymphoid tissues, making up maybe 10–20% of blood lymphocytes. Unlike T cells, B cells can recognize a much broader range of antigens in their native, unprocessed form proteins, polysaccharides, lipids, nucleic acids directly, without needing MHC presentation. This happens through the B cell receptor complex, the BCR.
And what makes up this BCR complex?
The main antigen-binding parts on a naive B cell are membrane-bound antibodies, specifically IgM and IgD isotypes. These are linked to signaling proteins called Ig-alpha, Ig-I, and Ig-beta, Ig-O. Ig-I and Ig-A have intracellular bits that kick off the activation cascade when antigen binds. There's also another molecule, CD21, a receptor for a complement component, C3d. Engaging CD21 provides a crucial costimulatory signal, helping B cell activation, especially if the antigen is coded with complement. Now, once a B cell meets its antigen and gets the right signals, often including help from CD4 plus T helper cells, it can differentiate into two main cell types: plasma cells and memory cells.
Plasma cells, those are the antibody factories, the really efficient ones.
Exactly. Highly specialized, terminally differentiated B cells. They just churn out antibodies hundreds, thousands per second. These antibodies circulate, neutralizing pathogens or marking them for destruction. The other type are memory B cells. Long-lived cells that have seen the antigen before, undergone some maturation. If they see the same antigen again, they reactivate much faster, more potently, often with higher affinity antibodies compared to the first time a naive B cell gets activated.
So we have these specialized lymphocytes constantly circulating. Where do these complex immune responses actually get started and coordinated?
The immune system has a very organized structure, primary and secondary lymphoid organs. The primary or generative ones are where mature lymphocytes are made and educated, bone marrow for B cells, thymus for T cells, where they learn self from non-self. The secondary or peripheral lymphoid organs are the strategic locations where immune responses usually kick off. These include lymph nodes, filtering lymph, the spleen filtering blood, and mucosal-associated lymphoid tissues, MALT, like tonsils, Peyer's patches in the gut guarding mucosal surfaces.
And this constant circulation you mentioned earlier, that's critical for immune surveillance across the whole body, isn't it?
Absolutely. Lymphocytes are always moving blood, lymph system, through lymphoid organs, other tissues. Naive T cells, for example, constantly cruise through secondary lymphoid organs like lymph nodes. They enter from the blood, look around for their specific antigen, and if they don't find it, they leave via the lymphatics. This recirculation massively increases the chance that the rare T cell specific for a new pathogen will actually encounter it. Once effector lymphocytes activated, functional cells are generated. They migrate out to the sites of infection. Plasma cells, though, tend to stay mostly in lymphoid organs and bone marrow, pumping antibodies into the circulation. This dynamic movement and organization ensures the system can respond anywhere. Plus, within these secondary organs, different cells are segregated into zones: B cells and follicles, T cells in the paracortex, which facilitates the interactions needed for an efficient response.
Okay, we've got the key cells and tissues down. Now, let's tackle this really crucial peptide display system, the major histocompatibility complex, MHC. It sounds like it's right at the heart of how adaptive immunity functions.
It is absolutely fundamental. MHC molecules are specialized protein complexes on cell surfaces whose main job is to display peptide antigens to T lymphocytes. Without MHC, T cells just wouldn't see invaders or abnormal self-proteins inside cells. And as we touched on, there are two main classes critical for T cell recognition: class I MHC and class I MHC molecules.
Let's start with class I MHC. Where do we find these in the body?
Class I MHC molecules are expressed on pretty much all nucleated cells in the body and also on platelets. Structurally, it's a heterodimer, two different chains linked together. One is the larger polymorphic alpha chain, encoded by HLA-A, B, and AC genes in the MHC locus. Polymorphic means there are many different versions or alleles in the population, making these chains highly variable between people. The other chain is smaller, non-polymorphic, called beta-2 microglobulin, BID miter, encoded outside the MHC locus. The alpha chain folds into three domains: IA1, IA2, IA3. The IA1 and IA2 domains together form a groove or cleft. That's where the peptide antigen binds. The polymorphic residues, the variable parts, are mostly in this cleft, which explains why different alleles bind different sets of peptides.
And where do the peptides displayed by class I MHC come from? What kind of proteins?
They come from proteins inside the cell, in the cytoplasm. This includes all the normal cellular proteins being made and broken down constantly, but crucially, it also includes proteins made by intracellular pathogens, like viruses replicating inside, or even abnormal proteins made by tumor cells due to mutations. These proteins are chopped up into peptides by machinery called proteasomes. Peptides of the right size, usually 8-11 amino acids, are transported into the endoplasmic reticulum, the ER. In the ER, they bind to newly made class IMHC heavy chains, which then link up with BET2 microglobulin. This stable complex then travels through the Golgi and finally to the cell surface, ready to be scanned by CD8 plus T cells.
You mentioned the alpha-3 domain of class IMHC is non-polymorphic. What's its specific role in recognition?
Right, the 83 domain has a specific binding site for the CD8 protein found on CD8 plus T cells. So when a CD8 plus T cell encounters a cell displaying a peptide class I complex that its TCR recognizes, the CD8 molecule acts as a co-receptor. It binds to this 83 domain, helping stabilize the interaction between the T cell and the target cell. More importantly, the CD8 binding also helps kick off the signaling inside the T cell needed for activation, and ultimately killing the target cell if it's deemed infected or abnormal. Because CD8 plus T cells are geared to recognize peptides on class IMHC, they're called class IMHC restricted, and since viruses or tumors can arise in almost any nucleated cell, it makes sense that all these cells express class IMHC for constant surveillance by CD8 plus CTLs.
Okay, very clear picture of class I. Now, class IMHC molecules, how do they differ expression, structure, peptide source?
Class II MHC molecules have a more restricted expression pattern. They're mainly found on specialized immune cells called antigen-presenting cells, or APCs. The main professional APCs are macrophages, B lymphocytes, and dendritic cells. The genes for class II MHC are in a different region of the MHC locus, called HLA-D, with subregions DP, DQ, DR. Each class II molecule is also a heterodimer, but it's made of two polymorphic chains, an alpha chain and a beta chain. Similar to class I, the peptide binding cleft in class II is formed by the interaction of the A1 domain and the BI1 domain. And again, polymorphism in these domains determines which peptides can bind.
And where do the peptides displayed by class II MHC come from? How are they loaded?
These peptides come from extracellular microbes and proteins that have been internalized by the APC through processes like phagocytosis or endocytosis. Inside the ATC, these proteins are broken down into peptides within acidic compartments like endosomes and lysosomes. These peptide-containing vesicles then fuse with other vesicles carrying newly synthesized class II MHC molecules. Peptide binding to class II MHC happens inside these vesicles. Special chaperone molecules help swap out a placeholder peptide called CLIP for the antigenic peptides. Once loaded, the peptide MHC class II complex travels to the cell surface, ready for recognition by CD4 plus T cells.
And just like CD8 interacts with class I, there must be a similar interaction for CD4 plus T cells in class II MHC.
Precisely. The beta-2 domain of the class II MHC beta chain has a binding site for the CD4 protein on CD4 plus T helper cells. When a CD4 plus T cell recognizes its specific peptide class II complex on an APC, the CD4 molecule acts as a co-receptor binding to this beta-2 domain. This helps stabilize the binding and, crucially, delivers essential activation signals into the CD4 plus T cell. This interaction is needed for the T cell to get activated and differentiate into helper cells that orchestrate the broader immune response against whatever extracellular threat the APC ingested. Because CD4 plus T cells specifically recognize antigens on class II MHC, they are class II MHC restricted.
This MHC system is incredibly diverse with all these HLA alleles. What's the significance of an individual's HLA haplotype?
The HLA haplotype is the specific combination of HLA alleles. Someone inherits one set from each parent for each major HLA gene, A, B, C, DP, DQ, DR. Because these genes are so polymorphic, so many different versions exist in the population, the chance of two unrelated people having the exact same haplotype is extremely low unless they're identical twins. This diversity is thought to be a huge evolutionary advantage for the population. It ensures that someone in the population will likely have MHC molecules capable of presenting peptides from almost any new pathogen that comes along. But this polymorphism is also the major hurdle in transplantation. The recipient's immune system sees the donor's different MHC molecules as foreign and attacks the graft. That's why HLA matching is so critical.
And you briefly mentioned class III MHC molecules. What are those? Are they involved in antigen presentation too?
That's a bit of a historical term. The MHC locus on chromosome 6 is packed with genes. Besides the class I and class II genes for antigen presentation, there is a region historically called class III. But the genes here don't encode antigen-presenting molecules like class I and II. Instead, they code for other important immune-related proteins, things like complement components, C2, C4, factor B, cytokines like TNF and lymphotoxin alpha, and other regulatory molecules. So they're in the neighborhood, genetically speaking, but have different functions.
Okay, got it. We now have a solid foundation in the normal immune response. Let's now dig into cell-mediated immunity, how T lymphocytes get activated, and how they eliminate intracellular threats.
Right. Cell-mediated immunity is primarily driven by T lymphocytes, especially CD4 plus helpers and CD8 plus cytotoxic T cells, CTLs. The activation process starts in secondary lymphoid organs like lymph nodes. Naive T cells, those that haven't met their antigen yet, need two key signals from antigen-presenting cells, most importantly dendritic cells, to get fully activated. Signal 1 is the specific recognition, the T cell receptor binding to its unique peptide MHC complex on the APC. That's the antigen-specific signal. But that signal alone usually isn't enough. It might even shut the T cell down. So a second signal, co-stimulation, is also required. This comes from specific co-stimulatory molecules on the APC binding to corresponding receptors on the T cell.
And what happens once a naive T cell gets both of those essential signals?
Once it gets both signals antigen via TCR and co-stimulation, it activates. This triggers internal signaling leading to several critical outcomes. Proliferation, where the cell divides rapidly, making lots of copies. Differentiation, where it matures into an effector cell ready to do its job, like secreting cytokines or killing. And the generation of memory cells for long-term immunity. These activated effector T cells then leave the lymphoid organs and travel to the specific sites where the triggering microbial antigens are located.
Let's focus first on the CD4 plus helper T cells. What are their initial actions and key functions once they become effector cells?
One of the earliest, most important things an activated CD4 plus helper T cell does is secrete a cytokine called interleukin-2, IL-2. At the same time, it ramps up expression of high-affinity IL-2 receptors on its own surface. This creates an autocrine loop. It makes IL-2 and responds to it, driving its own proliferation and differentiation. This self-stimulation is critical for rapidly expanding the number of specific helper T cells needed. Beyond that initial boost, the main functions of effector CD4 plus helpers are mediated by a surface molecule called CD40 ligand, CD40L, and the various cytokines they secrete.
How does CD40L help these T cells perform their helper function?
When a CD4 plus helper recognizes its antigen being presented by, say, a macrophage or a B cell, it expresses CD40L on its surface. This CD40L then binds to its receptor, CD40, which is on the macrophage or B cell. This binding delivers a crucial activating signal to the macrophage or B cell. For macrophages that have eaten microbes, this helps activate them to kill better. For B cells that have seen their antigen, this CD40L interaction is essential help for them to switch antibody classes from IgM to IgG, IgA, etc., and mature into high-affinity antibody-secreting plasma cells.
And the cytokines these CD4 plus cells secrete, they're incredibly diverse and really coordinate the whole response, don't they?
Absolutely. Cytokines are the immune system's communication network. These soluble proteins are secreted by various cells, including CD4 plus T cells, bind to specific receptors on target cells, and trigger a huge range of effects. Different subsets of CD4 plus effector T cells, which we'll get to, make distinct sets of cytokines tailoring the response. Some cytokines recruit other leukocytes, like neutrophils, others boost macrophage killing power, others promote B cell differentiation, some even suppress the response to prevent damage. Understanding cytokines has been huge for medicine. Think about TNF antagonists, drugs that block the cytokine TNF used successfully in diseases like rheumatoid arthritis, where TNF drives inflammation.
Right.
Conversely, we can sometimes give cytokines therapeutically, like GCSF to boost neutrophils, or interferons to fight viruses, or even cytokines to mobilize stem cells for transplantation.
We can't forget the CD8 plus cytotoxic T lymphocytes, CTLs. How do they get activated into killers, and how do they actually eliminate infected or cancerous cells?
Similar to CD4 plus cells, precursor CD8 plus T cells also need activation in lymphoid organs by recognizing their specific peptide MHC class I complex on an APC, often a dendritic cell. They also need co-stimulation, sometimes requiring help from activated CD4 plus cells for full function. Once activated into effector CTLs, they gain the critical ability to recognize and kill other body cells, expressing foreign antigens in their cytoplasm presented on MHC class I. This is crucial for eliminating virus-infected cells or tumor cells displaying abnormal proteins. CTLs use several effective methods to kill targets. One main way is releasing cytotoxic granules. These contain proteins like perforin, which post holes in the target cell membrane. Through these holes, granzymes enzymes enter the target cell. Granzymes directly activate apoptotic pathways, causing the target cell to self-destruct in a controlled way, preventing pathogen release. CTLs can also express a surface protein called Fos ligand, FOS-SL. If the target cell expresses the Fos receptor, Fos-L binding triggers a different apoptotic pathway, also leading to cell death. Plus, besides killing, some CTLs can also produce cytokines like interferon gamma, IFN now, and TNF, contributing to inflammation and having direct antiviral or anti-tumor effects.
You've mentioned dendritic cells a few times as key APCs for activating T cells. What makes them so special for this job?
Dendritic cells, or DCs, are really the sentinels. They're positioned in tissues like skin and mucosa, where they are incredibly good at capturing antigens from invading microbes. Once they grab an antigen, they undergo maturation. They start migrating from the tissues to the draining lymph nodes. As they travel, they dramatically ramp up their expression of both MHC class I and class II molecules, becoming loaded with peptides. And crucially, they also significantly increase expression of those costimulatory molecules, like B7 proteins, CD80, CD86. So by the time they reach the lymph node, they are fully equipped to effectively present antigens to naive T cells and deliver the essential second signals needed for activation into effector and memory cells. They are the vital bridge between innate sensing of danger and initiating the specific adaptive response. Their unique ability to activate naive T cells makes them essential for starting almost all primary adaptive immune responses.
And we can't overlook the different flavors of CD4 plus helper T cells, Th1, Th2, Th17. What determines which path a naive CD4 plus cell takes, and what are their distinct jobs?
Right. When a naive CD4 plus T cell gets activated, the specific cytokines present in the local environment at that time play a key role in directing its differentiation into one of these specialized subsets, Th1, Th2, or Th17. Each subset then makes its own characteristic set of cytokines, mediating different types of immune responses effective against different kinds of pathogens. For example, Th1 differentiation is usually driven by cytokines like IFN and IL-12, often produced early in response to intracellular pathogens. Th1 cells then mainly produce IFN themselves, which is great at activating macrophages to kill ingested microbes, crucial for cell-mediated immunity against intracellular bacteria and viruses.
Okay, Th1 for intracellular stuff.
The H2 differentiation is often driven by IL-4, frequently seen in response to parasites like worms and also in allergic reactions. Th2 cells make cytokines like IL-4, IL-5, and IL-13. These orchestrate responses against extracellular parasites, promoting IgE antibody production for mast cell eosinophils and activating eosinophils. Th17 differentiation needs cytokines like TGF-thick, IL-6, and IL-23. Th17 cells produce IL-17 and IL-22, which are important for defense against extracellular bacteria and fungi, especially at mucosal surfaces. They do this by recruiting neutrophils and boosting antimicrobial peptide production by epithelial cells. Now, this is a bit simplified. Reality is more complex. Some T cells make multiple cytokines. There's plasticity between subsets, but it's a useful framework.
And finally, let's circle back to memory cells. They're the key to long-lasting protection after infection or vaccination, aren't they? What makes them so effective on round 2?
Exactly. The initial activation doesn't just make effector cells for the current fight. It also creates a pool of long-lived memory lymphocytes specific for that antigen memory T cells, CD4 plus and CD8 plus, and memory B cells. These memory cells have several advantages over naive cells. First, there are usually more of them specific for that antigen. Second, they often express different surface molecules, allowing them to home more efficiently to infection sites. Most importantly, they're functionally primed to respond much faster and more robustly upon re-exposure. Memory T cells often need weaker activation signals and differentiate into effectors quicker. Memory B cells have usually undergone affinity maturation, making higher affinity antibodies, and they rapidly become plasma cells, often switching isotypes faster than in the primary response.
Right, so they're faster and stronger.
Precisely. This rapid enhanced response is why vaccination works. We expose the immune system to a safe version of the pathogen or its parts, generating these memory cells. Then, if you encounter the real pathogen later, these pre-existing memory cells mount a swift, powerful response, often preventing disease entirely or making it much milder.
So that gives us a really comprehensive understanding of how our bodies normally fight threats. Now let's shift focus to when this finely tuned system goes wrong, starting with hypersensitivity disorders, when the immune system overreacts to things that are usually harmless.
Right. Hypersensitivity reactions are basically immune responses that end up causing tissue injury. They happen when the normal protective mechanisms become inappropriately activated, are too intense, or get misdirected against innocuous things like environmental allergens, or even against the body's own self-antigens. In many cases, it's thought that a failure in the normal regulatory mechanisms, the checks and balances, is a key underlying factor.
And these disorders often have a genetic component. Some people seem more prone.
Yes, there's a significant link between genetics, particularly certain HLA alleles, and the risk of developing specific hypersensitivity diseases, including allergies and autoimmune conditions. Many non-HLA genes involved in immune regulation have also been implicated. We'll see specific examples as we go.
So the actual mechanisms causing damage and hypersensitivity are often the same tools the immune system uses to fight infection. The problem is they're being used inappropriately, wrong target, wrong intensity.
Exactly. Antibodies, CTLs, macrophages, inflammatory mediators, all the usual weapons end up damaging our own tissues. These reactions are broadly classified into four main types, type I through 4, based mainly on the immune mechanisms involved.
Let's break these down. First, type I, immediate hypersensitivity. Key players and sequence of events.
Type I is driven by Th2 helper T cells, IgE antibodies, and mast cells, though basophils and eosinophils also play roles, especially later. It starts with initial exposure to an allergen pollen, dust mites, food proteins in a susceptible person. This activates Th2 cells, which release cytokines like IL-4, telling B cells to switch to making IgE antibodies specific for that allergen. This IgE then circulates and binds tightly to specific FUV receptors, FCRI, on mast cells and basophils. Now, these cells are sensitized.
Okay, coded with IgE.
Right. Then, upon re-exposure to the same allergen, the allergen molecules cross-link the IgE already bound to these receptors. This cross-linking triggers rapid degranulation, the release of pre-stored mediators like histamine, protease, etc. These mediators cause the immediate reaction within minutes. Vasodilation, leaky vessels, smooth muscle contraction, like in airways, mucus secretion. Think allergy symptoms. This is often followed hours later, 2 to 24 hours, by a late phase reaction. This involves recruiting other inflammatory cells, eosinophils, neutrophils, lymphocytes, which release more mediators, causing sustained inflammation and more tissue damage.
And typical examples.
Systemic anaphylaxis, severe whole body reaction, allergic asthma, allergic rhinitis, hay fever, food allergies. Those are classic type I disorders.
Okay, so sensitization, IgE, mast cell degranulation. What about type 2, antibody-mediated hypersensitivity? How does that cause injury?
In type 2, tissue injury is mainly caused by IgG and IgM antibodies directed against antigens on cell surfaces or in the extracellular matrix. They cause damage in several ways. One is promoting phagocytosis. Antibodies coat the target cell, opsonization, and phagocytes grab onto the antibody Fc region or bound complement like C3b and eat the cell. Another is complement activation directly on the cell surface by the bound antibody. This can lead to formation of the membrane attack complex, MAC, punching holes in the cell membrane and causing lysis, especially effective against thin-walled cells like red blood cells.
And sometimes they just interfere with function?
Exactly. Antibodies can bind to cell surface receptors or essential molecules and block or inappropriately stimulate their normal function without directly killing the cell. Examples of type 2 include transfusion reactions, hemolytic disease of the fetus and newborn, autoimmune hemolytic anemia, thrombocytopenia, low platelets, some drug reactions where the drug sticks to cells, and those functional interference examples. Myasthenia gravis, antibodies block acetylcholine receptors, Graves' disease, antibodies stimulate the TSH receptor causing hyperthyroidism, pernicious anemia, antibodies block intrinsic factor needed for B12 absorption. Sometimes antibodies also trigger inflammation by activating complement and recruiting leukocytes that release damaging enzymes.
Next, type 3, immune complex mediated. What's the core process here?
Type 3 involves antigen-antibody immune complexes, usually IgG or IgM binding to antigens, often soluble ones circulating in the blood. The key problem is the deposition of these preformed complexes in tissues, vessel walls, kidney glomeruli, joints, especially where there's high filtration pressure. Once deposited, these complexes activate complement. This generates C3a and C5a, which attract inflammatory cells, mostly neutrophils. These activated neutrophils arrive and try to clear the complexes, but end up releasing enzymes and reactive oxygen species that damage the surrounding tissue.
Okay, so the complexes deposit, then cause inflammation.
Right. A hallmark is necrotizing vasculitis inflammation and necrosis of blood vessel walls. Classic examples are systemic lupus erythematosus, SLE, some types of glomerulonephritis, serum sickness, reaction to foreign proteins, and the Earthist reaction, a localized skin reaction. Serum sickness is the prototype systemic complex disease, antibody response, complex formation, deposition, then widespread inflammation. The Arthus reaction is localized. Inject antigen into someone with high antibody levels, complexes form locally in vessel walls, causing local vasculitis and necrosis.
And finally, type IV, cell-mediated hypersensitivity. This one's different T cells, not antibodies. What are the main ways T cells cause injury here?
Correct. Type IV is mediated by activated T lymphocytes, CD4 plus helpers, and CD8 plus CTLs. Two main mechanisms. First is delayed type hypersensitivity, or DTH. This is mainly driven by CD4 plus Th1 cells. They recognize antigen presented by APCs in tissues and release cytokines, especially interferon gamma, IFN-NUC. IFN potently activates macrophages. These activated macrophages release inflammatory mediators, enzymes, ROS, causing tissue damage. If activation is sustained, macrophages can form epithelioid cells and aggregate into granulomas trying to wall off persistent antigens, like in tuberculosis. This response is delayed, taking 24-72 hours.
Okay, Th1 activating macrophages leads to damage. What's the second mechanism?
The second is direct T-cell mediated cytotoxicity, driven by CD8 plus CTLs. These CTLs directly recognize and kill target cells expressing specific antigens on MHC class. I like virus-infected cells or tumor cells. This is also key in transplant rejection. Examples of type IV include contact dermatitis, like poison ivy, multiple sclerosis, T cells attack myelin, type 1 diabetes, T cells destroy pancreatic beta cells, and the tissue damage in tuberculosis.
So we've seen how the immune system can overreact. Now let's shift to another major problem: autoimmune diseases, where the immune system mistakenly targets the body's own tissues.
Exactly. Autoimmune diseases result from a fundamental failure of self-tolerance, the normal mechanisms that stop our immune system attacking our own cells and tissues.
Wow.
Because tolerance breaks down, immune responses involving both antibodies and T cells develop against self-antigens, leading to chronic inflammation and damage in various parts of the body. It's usually a complex mix of inherited genetic susceptibility and environmental triggers, like infections or tissue damage.
What are some key reasons this critical self-tolerance might fail?
Several potential breakdowns. One is in central tolerance during lymphocyte development. In the thymus, T cells strongly reactive to self-antigens should be deleted or turned into regulatory T cells. If this fails, autoreactive T cells escape. Similarly, in the bone marrow, autoreactive B cells should be eliminated or inactivated. Then there's peripheral tolerance, acting on escaped autoreactive cells in the periphery. This includes anergy, functional unresponsiveness due to lack of co-stimulation, suppression by regulatory T cells, and deletion via activation-induced cell death. Failures here are critical.
You mentioned regulatory T cells, TREGs. They're like the immune system's internal police, keeping things in check.
That's a good analogy. TREGs, mostly CD4 plus cells expressing the FOXP3 transcription factor, are crucial for maintaining balance and preventing autoimmunity. They actively suppress other immune cells, including self-reactive ones. They use various methods like secreting immunosuppressive cytokines, IL-10, TGF-O, or direct cell contact involving molecules like CTLA-4. Defects in TREG development or function are strongly linked to autoimmunity. Mutations in FOXP3 cause the severe IPEX syndrome. IL-2 is vital for TREG maintenance, so defects in IL-2 or its receptor also cause autoimmunity. Polymorphisms affecting CD25, part of the IL-2 receptor, are also linked to autoimmune risk, suggesting TREG dysfunction plays a role.
Another potential trigger was abnormal display of self-antigens. How does that contribute?
Normally, some self-antigens might be hidden away, sequestered. If tissue injury or inflammation causes increased release or persistence of these antigens, or if they get structurally altered, maybe post-translational modifications like citrullonation, or due to cell stress, they might appear as neoantigens. The immune system hasn't been tolerized to these altered forms and might mount a response, kicking off autoimmunity.
And genetics seems to play a big role in susceptibility.
Yes, most autoimmune diseases are complex polygenic disorders. Multiple genes contribute. We see higher concordings in identical twins than fraternal twins. HLA genes within the MHC have the strongest associations. Ankylosing spondylitis is strongly linked to HLA-B27. Many others, like RA, type 1 diabetes, MS, are linked to specific class II HLA alleles, DR, DQ. The thinking is that certain HLA molecules might be better at presenting specific self-peptides, triggering autoreactive T cells, but having the gene doesn't guarantee disease, it just increases risk. Genome-wide studies have also found many non-MHC genes associated with risk, often involved in immune regulation pathways, highlighting the complexity.
What about infections and other environmental factors? Can they really trigger autoimmunity?
There's compelling evidence they can in genetically susceptible people. One mechanism is infection upregulating costimulatory molecules on APCs. If these APCs are also presenting self-antigens, it might override anergy and activate self-reactive T cells. Another is molecular mimicry. A microbe might have an antigen that looks very similar to a self-antigen. The immune response to the microbe might then cross-react with the self-antigen. Rheumatic heart disease after strep infection is a classic example. Antibodies against strep proteins cross-react with heart muscle.
It's interesting.
Paradoxically, there's also the hygiene hypothesis, the idea that fewer infections and cleaner environments might actually increase risk for some autoimmune or allergic diseases, perhaps by affecting TREG development. And tissue injury itself can expose self-antigens and activate local immune cells, potentially initiating autoimmunity.
Okay, with those general principles, let's dive into specific systemic autoimmune diseases, starting with one that seems to affect almost everything. Systemic lupus erythematosus, SLE.
Right. SLE is a chronic multi-system autoimmune disease. It's characterized by a huge array of autoantibodies, especially anti-nuclear antibodies, ANAs, targeting components of the cell nucleus. Tissue injury is mainly from immune complex deposition, type 3 hypersensitivity, and also direct antibody binding to cells and tissues, type 2. Clinically, it's very heterogeneous, acute or insidious onset, chronic, relapsing, remitting course, often with fever, fatigue. Skin, joints, kidneys, serosal membranes, pleura, pericardium are commonly hit hard, but really any organ can be affected. Because it's so diverse, there are classification criteria, things like malar rash, discoid rash, photosensitivity, oral ulcers, arthritis, serositis, kidney problems, neurological issues, blood cell abnormalities, specific autoantibodies, and a positive ANA test. You need a certain number to meet the classification for research.
And the range of autoantibodies in SLE is huge, right? Tell us more about ANAs and others?
Yeah. Yes, it's remarkable. ANAs are present in almost all SLE patients, making them a sensitive marker, though not perfectly specific. They target nuclear stuff. DNA, double-stranded and single-stranded, histones, RNA-binding proteins like SM antigen RNPs, nucleolar components. We usually detect ANAs using indirect immunofluorescence on cells. The pattern of staining homogeneous, rim, speckled, nucleolar centromeric gives clues about the specific antibodies present.
Patterns give clues.
Exactly. For instance, antibodies to double-stranded DNA, anti-dsDNA, and the SEP antigen, anti-charism, are highly specific for SLE and part of the criteria. Besides ANAs, SLE patients often make antibodies against blood cells, causing anemia, low platelets, etc., and also antiphospholipid antibodies. These bind phospholipids or associated proteins. Antiphospholipid antibodies are clinically important because they increase the risk of blood clots, miscarriages, sometimes called antiphospholipid syndrome. Paradoxically, one test, the lupus anticoagulant, prolongs clotting time in vitro but causes clotting in vivo. Some can also cause a false positive syphilis test. Certain HLA alleles, especially in the HLA-DQ locus, slightly increase risk and are linked to specific antibodies like anti-dsDNA. Inherited deficiencies of early complement components, C1q, C4, C2, are rare but strongly associated with SLE, possibly because they impair clearance of immune complexes and apoptotic debris. Historically, LE cells, phagocytes that ate antibody-coated nuclei were used for diagnosis, but that's mostly replaced by specific antibody tests now.
So how do these antibodies and immune complexes cause all that widespread tissue damage in SLE?
Most systemic damage is thought to be from immune complex deposition type 3 hypersensitivity. DNA anti-DNA complexes depositing in kidney glomeruli and small vessels is a key mechanism seen in models in patients. Low serum complement levels during active disease, because it's being used up by complexes, and finding immunoglobulin and complement deposits in biopsies, like granular deposits in kidney glomeruli, strongly support this.
Okay, mainly immune complexes.
Primarily, yes, but T cells are also found infiltrating affected tissues like the kidneys, suggesting cell-mediated mechanisms also contribute to the damage. The overall model involves genetic susceptibility, environmental triggers like UV light, viruses, leading to persistent nuclear antigens. This drives autoantibody production. The response gets amplified by nucleic acids stimulating TLRs on DCs and B cells, and by type 1 interferons contributing to the chronic inflammation.
Given SLE's wide reach, what are some characteristic changes pathologists see in affected tissues?
It's variable, but common patterns involve immune complex deposition in blood vessels, kidneys, connective tissue, skin. Lupus nephritis, kidney involvement is very common, 50–70%. There are different classes of lupus nephritis histologically. Class 4, diffuse proliferative, is severe. You see proliferation of kidney cells, immune complex deposits under the endothelium looking like wire loops on microscopy, and granular Ig complement deposits on immunofluorescence. This can scar the kidney, clinically. Blood protein in urine, high blood pressure, kidney failure.
What about other organs?
Joints often show non-erosive synovitis inflammation without major bone disruption, unlike RA. CNS involvement causing neuropsychiatric symptoms often lacks clear structural changes, maybe some small vessel occlusion. Serositis inflammation of linings like pericardium, pericarditis, or pleura, pleuritis is common, causing inflammation fluid, sometimes scarring. The heart can have pericarditis, myocarditis, or Libman-Sachs endocarditis, small vegetations on valves. Skin involvement is huge, 85%. The classic butterfly malar rash, discoid rash, photosensitivity, ulcers, immunofluorescence of skin shows IgC3 deposits at the dermal epidermal junction, the lupus band test.
You also mentioned chronic discoid lupus. How's that different from systemic lupus?
Chronic discoid lupus, DLE, primarily affects the skin, mimicking some SLE skin signs like the discoid rash, but systemic organ involvement is rare. DLE lesions are typically well-defined, raised red plaques, often scaly, with follicular plugging, that tend to heal with scarring and atrophy, common on sun-exposed areas. While usually confined to skin, a small percentage, 5–10%, might eventually develop systemic SLE. About a third might have a positive generic ANA test, but anti-dsDNA antibodies are rare. Skin biopsy immunofluorescence looks similar to SLE lesions.
Let's move to another systemic autoimmune disease, Sjogren's syndrome, primary features and underlying immunology.
Sjogren's syndrome is chronic, systemic, characterized by immune-mediated destruction of lacrimal and salivary glands, leading to the classic dry eyes, keratoconjunctivitis sicca, and dry mouth, xerostomia. It can be primary, Sicca syndrome alone, or secondary, associated with other autoimmune diseases. RA is the most common, but also SLE, systemic sclerosis, etc. It predominantly affects women, peaking around age 50–60. Symptoms stem from exocrine gland destruction, gritty eyes, blurred vision, difficulty swallowing dry food, dry mouth, fissures. About half get parotid gland enlargement. Dryness can affect other mucosa too, nosebleeds, airways, bronchitis. About a third get extraglandular issues like synovitis, lung fibrosis, neuropathy, especially if they have high anti-SSA-RO antibodies. Kidney problems are less common than in SLE, but tubular issues can occur. Importantly, there's a significantly increased risk of B-cell lymphomas.
What do the affected glands look like morphologically in Sjogren's?
Lacrimal and salivary glands are the main targets. Early on, histology shows lymphocytes infiltrating around ducts and vessels. Later, the infiltrate becomes extensive, often forming lymphoid follicles with germinal centers right in the gland. Ductal cells might proliferate, blocking ducts. Eventually, there's acinar atrophy, fibrosis, and fatty replacement. The infiltrate can sometimes be so dense it mimics lymphoma. Lack of tears leads to corneal drying, inflammation, ulcers. Oral mucosa can atrophy and fissure. Nasal dryness can cause crusting, even septal perforation.
What's the current thinking on the pathogenesis? What drives the gland destruction?
It involves lymphocytic infiltration and fibrosis. The infiltrate is mostly activated CD4 plus T cells, but also B cells and plasma cells, sometimes forming those germinal centers, suggesting local antibody production. Many patients, 75%, have rheumatoid factor, and 50–80% have ANAs. The most characteristic antibodies, though, are against ribonucleoprotein antigens SSA-RO and SSB-LA, found in up to 90% of primary cases. While diagnostic markers, their direct role in damage isn't fully clear. The leading hypothesis is an autoimmune T-cell reaction against unknown self-antigens in the glands, or maybe an immune reaction to a virus infecting the glands. Over time, dominant B-cell clones can emerge within the inflamed glands, showing signs of antigen-driven selection. This clonal expansion is thought to underpin the much higher risk, around 40-fold, of developing marginal zone lymphoma. Other autoimmune diseases targeting organs, like Hashimoto's thyroiditis, also carry higher lymphoma risk in the target tissue. The specific triggers and self-antigens are still under investigation.
Let's turn now to systemic sclerosis, or scleroderma, defining characteristics and tissues affected.
Systemic sclerosis is chronic, progressive, characterized by widespread fibrosis, excessive connective tissue buildup in skin, GI tract, and other organs, along with vasculopathy or blood vessel disease. The pathogenesis is complex, involving genetics, environment, immune activation, and microvascular abnormalities, all leading to excessive collagen deposition.
What's the current model for how this unfolds?
The idea is that in susceptible people, some unknown trigger starts things off, causing both vascular issues and immune activation. These seem interconnected and feed each other, ultimately activating fibroblasts too much, causing fibrosis. Both T and B cells get activated, producing profibrotic cytokines like TGF-4, IL-13, PDGF, which stimulate fibroblasts to make collagen. Autoantibodies like ANAs, Anti-serral 70, topoisomerase I, and anti-centromere antibodies are common and useful for diagnosis prognosis, but their direct role in causing fibrosis is less clear than in some other diseases. Some speculate they might stimulate cells, but evidence is weaker. Vascular damage, though, especially microvascular disease, is a consistent early feature. You see intimal thickening in digital arteries, capillary dilation, leakage, and destruction visible in nail fold capillaries early on. There's endothelial injury and activation, platelet activation.
So the vessel damage is key.
It seems central. Repeated endothelial injury, maybe from inflammation or antibodies, plus platelet aggregation, releases growth factors like PDGF, TGFA, driving perivascular fibrosis. Smooth muscle abnormalities might contribute to Raynaud's. Widespread microvessel narrowing leads to ischemia and scarring in multiple organs. The exact cause of the initial vascular injury is still debated. Is it the trigger or a result of chronic inflammation?
Which organs get hit hardest in systemic sclerosis, and what do they look like?
Almost any organ can be involved, but severity varies. Skin shows progressive sclerotic atrophy, starting in fingers, moving up the arms, face. Histology. Early on, T-cell infiltrates and edema. Later, dense collagen in the dermis, thin epidermis, loss of hair, folliculous wet glands. The GI tract, especially the lower esophagus, gets fibrosed, leading to poor motility, dysphagia. Intestines can have fibrosis causing malabsorption. Musculoskeletal. Early synovitis, later joint fibrosis, less destructive than RA. Inflammatory myositis in about 10%. Kidneys are serious. Vascular lesions in about 2/3, mainly intimal thickening of small arteries. This can lead to hypertension in 30%, and about 20% get malignant hypertension with rapid renal failure, a major cause of death. Specific glomerular changes aren't typical. Lungs often get interstitial fibrosis, honeycomb lung, and pulmonary vascular sclerosis can cause pulmonary hypertension.
What clinical features would make you suspect systemic sclerosis?
The striking skin changes, thickening, tightening are key. Raynaud phenomenon is almost universal and often the first sign, appearing months or years before other symptoms. Dysphagia from esophageal issues is very common, 50%. Other GI issues like pain, obstruction, malabsorption, lung symptoms like shortness of breath, cardiac issues like pericardial effusion, fibrosis.
And those autoantibodies, anti-self-70, anti-centromere, what do they typically indicate?
Almost all patients have ANAs. Anti-self-70, anti-topoisomerase I is highly specific for the diffuse form of systemic sclerosis found in 10–20%. It's linked to faster progression, more lung fibrosis, peripheral vascular disease. Anti-centromere antibodies in 23% are characteristic of the limited cutaneous form, often called Crest syndrome, calcinosis, Raynaud's, esophageal dysmotility, sclerodactyly, telangiectasias. These patients tend to have a slower course, less early organ damage, better prognosis overall.
You briefly mentioned IgG4-related disease. Could you clarify what that is?
Right. IgG4-related disease is a more recently recognized condition. It's a fibroinflammatory disorder. The key features are tissue infiltrates, packed with IgG4-positive plasma cells, fibrosis often in a specific store form or cartwheel pattern, and often high serum IgG4 levels. It's systemic, affecting multiple organs, pancreas, autoimmune pancreatitis, bile ducts, sclerosing cholangitis, salivary lacrimal glands, mimicking Sjogren's, kidneys, lungs, retroperitoneum. The histology helps distinguish it from other fibrotic conditions like scleroderma. It's important because it often responds well to steroids.
We've covered autoimmunity and hypersensitivity extensively. Now let's flip to the other side. Transplantation immunology, where the immune system is the major obstacle to success.
Exactly. Rejecting allograft grafts from a genetically different person of the same species is a huge challenge. The recipient's immune system sees the donor tissue as foreign and attacks it. These responses against allografts are typically much stronger than responses to pathogens, mainly because there's a higher frequency of recipient T cells that can recognize the many genetic differences, especially MHC molecules between donor and recipient.
What are the different types of rejection after a transplant? Timelines, mechanisms?
Generally 3 main types: hyperacute, acute, and chronic. Hyperacute rejection happens within minutes to hours. It's antibody-mediated, occurring in recipients with pre-existing antibodies against donor HLA or ABO blood group antigens. These antibodies bind to the graft's vascular endothelium, activate complement, causing widespread blood clots, thrombosis, and graft death, infarction.
Okay, that's immediate. What about acute?
Acute rejection typically occurs days to weeks later, or later if immunosuppression fails. It's primarily mediated by T cells, CD4+ and CD8+, activated by the graft's foreign antigens, especially MHC. Antibodies newly formed against the graft also contribute, causing acute antibody-mediated humoral rejection. Most acutely rejecting grafts show both cellular and humoral patterns. Acute cellular T-cell rejection involves T cells infiltrating the graft. CD8 plus CTLs kill graft cells directly. CD4 plus helpers secrete cytokines causing inflammation. In kidneys, this can look like interstitial inflammation and tubulitis, type I, or inflammation of blood vessel linings, endothelialitis, type 2 vascular rejection. Acute antibody-mediated humoral rejection happens when new anti-donor antibodies damage graft blood vessels, especially glomeruli and peritubular capillaries. You see inflammation, capillaritis, and often deposition of complement C4d in these capillaries, a key diagnostic marker.
And chronic rejection.
Chronic rejection develops over months to years, a major cause of late graft failure. It's complex, involving immune and non-immune factors. A key feature is chronic allograft vasculopathy or graft arteriosclerosis, thickening and narrowing of graft blood vessels due to smooth muscle proliferation and matrix deposition. This is thought to result from chronic endothelial injury by T cells and antibodies. It leads to chronic ischemia, fibrosis, and eventual graft failure. Chronic antibody-mediated rejection also contributes, especially in kidneys, causing transplant glomerulopathy and often associated with C4d deposition.
You also mentioned graft versus host disease, GVHD. How's that different and where does it occur?
GVHD is unique to hematopoietic stem cell, HSC transplants, like bone marrow transplants. Here, it's not the recipient rejecting the graft, but the donor's T cells within the graft attacking the recipient's tissues as foreign. This happens because the recipient's immune system is usually wiped out beforehand. Acute GVHD, days, weeks, hits skin, rash, liver jaundice, GI tract, diarrhea. Chronic GVHD, months, years, causes fibrosis in skin, liver, lungs, GI tract. Interestingly, these donor T cells can also have a beneficial graft versus leukemia GVL effect, killing residual cancer cells. Removing T cells from the graft stops GVHD but increases relapse risk and infection risk, like EBV lymphoma, so it's a tricky balance.
Given these strong rejection responses, what kind of immunosuppression is needed?
Immunosuppression is essential for allografts, except identical twins. Combinations of drugs are used to target different immune pathways. Common agents include corticosteroids, broad anti-inflammatory, mycophenolate Mofetil inhibits lymphocyte proliferation, and calcineurin inhibitors like tacrolimus or cyclosporine block IL-2 production, key for T-cell response. Other tools include T-B cell depleting antibodies, pooled intravenous immunoglobulin, IVIG, which has complex suppressive effects, and plasmapheresis to remove antibodies in severe humoral rejection. Newer strategies aim to block stimulatory signals between APCs and T cells. The goal is enough suppression to prevent rejection but minimize side effects like infection and cancer risk.
We've covered the immune system overreacting or misdirecting its attack. Now let's turn to the opposite problem. Immunodeficiency diseases, where the system isn't strong enough.
Right. Immunodeficiency diseases mean the immune system can't fight off infections properly. This leads to increased susceptibility, more frequent, more severe infections, or infections by opportunistic organisms that usually don't cause trouble. We classify them as primary, congenital, caused by genetic defects affecting innate or adaptive immunity, or secondary acquired, resulting from other conditions like HIV, malnutrition, cancer, or immunosuppressive drugs. Studying primary immunodeficiencies has taught us a huge amount about how the normal immune system works.
Let's start with primary immunodeficiencies affecting innate immunity. Key categories and examples.
Defects can hit phagocytes, NK cells, or complement. For phagocytes, there is leukocyte adhesion deficiency type 1, LAD1, caused by defects in beta-2 integrins needed for leukocytes to stick to endothelium and get into tissues. This leads to recurrent bacterial fungal infections. LAD2 affects a different adhesion molecule, ligand, sialyl X. Similar result. Chronic granulomatous disease, CGD, involves defects in the NADPH oxidase enzyme complex in phagocytes. They can't make reactive oxygen species needed to kill ingested microbes. Result, recurrent bacterial fungal infections and granuloma formation because the microbes persist. Chidiac-Kagashi syndrome is a rare defect in lysosomal trafficking, impairing phagocyte function, NK cell killing, and also causing partial albinism and neurological issues.
And inherited defects in the complement system.
Yes, various complement deficiencies exist. C3 deficiency increases susceptibility to many bacterial infections. Deficiencies in the late components, C5-C9, needed for the membrane attack complex, MAC, make people particularly prone to Neisseria infections, meningitis, gonorrhea. C1 inhibitor deficiency causes hereditary angioedema with recurrent severe swelling due to uncontrolled activation of other systems. Deficiencies in complement regulatory proteins can lead to conditions like PNH or atypical HUS.
Now, primary immunodeficiencies affecting adaptive immunity T and B cells, these often have more profound effects.
Yes, because adaptive immunity is so central and T cells help B cells. Defects often impair both humoral antibody and cellular T cell responses. They can arise from problems in lymphocyte development, maturation, or activation. One major category is severe combined immunodeficiency, SCID.
SCID sounds very serious. Key characteristics and clinical picture.
SCID isn't one disease, but a group of distinct genetic syndromes, all resulting in profound defects in both T cell and B cell immunity. Infants typically present very early with severe, persistent infections like thrush, bad diaper rash, failure to thrive. They're extremely vulnerable to opportunistic infections, candida, pneumocystis, bacteria, pseudomonas, viruses, CMV, varicella. Even live vaccines are dangerous. Without a hematopoietic stem cell transplant to rebuild their immune system, classic SCID is usually fatal within the first year. The underlying genetic defects vary, but all lead to a severe lack of functional T cells, often with secondary B cell failure due to lack of T cell help.
What specific genetic defects cause SCID?
Many different causes. The most common is X-linked SCID, about 50%, caused by mutations in the common gamma ART chain gene. This protein is part of receptors for several key cytokines, including IL-7, which is essential for T cell development. So no functional ART chain means very few T cells and NK cells develop. B cells might be present, but don't work well without T cell help. Autosomal recessive SCID makes up the other half. About 50% of these are due to adenosine deaminase ADA deficiency. Lack of ADA leads to buildup of toxic metabolites that kill developing lymphocytes. ADA deficiency can be treated with enzyme replacement or gene therapy. Other causes include mutations in ART genes needed for rearranging antigen receptor genes, blocking T and B cell development, or mutations in JAK3 kinase, which signals downstream of the GOG chain receptor, causing a similar phenotype to X-linked SCID.
Let's discuss a primary B cell deficiency, X-linked agamaglobulinemia, or BRUTINS. Key features.
X-linked agamaglobulinemia, XLA, is characterized by a near absence of mature B cells and thus very low levels of all antibody classes. IgG, IgA, IgM, IgE. It's caused by mutations in the Bruton tyrosine kinase BTK gene on the X chromosome. BTK is crucial for signaling during B cell development, specifically for pre-B cells to mature. Without functional BTK, B cell development halts at the pre-B stage. Being X-linked, it mainly affects males. Maternal IgG protects infants for the first 6-9 months, then symptoms appear. The hallmark is recurrent bacterial infections, especially respiratory ones, caused by encapsulated bacteria normally cleared by antibodies, Haemophilus, Strep pneumo, Staph aureus. They're also susceptible to certain viruses like enteroviruses can cause chronic meningitis, and the parasite Giardia, but T-cell immunity is intact, so they handle most fungal and intracellular viral infections okay.
What about DiGeorge syndrome? That affects T-cell development due to thymus problems.
Yes, the George syndrome results from poor development of the thymus and parathyroid glands during embryogenesis, usually due to a deletion on chromosome 22q11.2, affecting the third and fourth pharyngeal pouches. This leads to variable T-cell deficiency, hypoplastic or absent thymus, hyperparathyroidism, low calcium, tetany, congenital heart defects, and distinct facial features. The T-cell deficiency severity varies greatly. Those severely affected have poor cell-mediated immunity prone to fungal viral infections. Lymphoid tissues lack T-cells. Antibody levels can also be variable depending on the T-cell defect.
You also mentioned hyper-IgM syndrome. What's the core issue there?
Hyper-IgM syndrome is a group of disorders with normal or high IgM levels but very low IgG, IgA, and IgE. This reflects a defect in immunoglobulin class switching. The most common form is X-linked, caused by mutations in the CD40 ligand CD40L gene on T helper cells. CD40L normally binds CD40 on B cells, providing a critical signal for class switching and affinity maturation. Without functional CD40L, B cells get stuck making only IgM. CD40-CD40L interaction also activates macrophages, so defects impair that too. Autosomal recessive forms can involve mutations in CD40 itself or in the 8 enzyme also needed for class switching. Patients get recurrent pyogenic bacterial infections, low IgG, pneumocystis pneumonia, defective macrophage activation, and sometimes autoimmune problems.
What about selective IgA deficiency? That sounds relatively common.
It is the most common primary immunodeficiency, 1 in 500 Caucasians, defined by low serum and secretory IgA, but normal IgG and IgM. Most people are actually asymptomatic. But IgA is key for mucosal defense, gut, airways. Symptomatic individuals may have more respiratory, GI, or urogenital infections. There's also a higher rate of allergies and autoimmune diseases like SLE-RA for reasons not fully clear. An important point. They can have severe anaphylactic reactions to blood transfusions containing normal IgA if they've developed anti-IgA antibodies.
Briefly, X-linked lymphoproliferative disease. Key defect.
XLP is a rare disorder with a dysregulated immune response to Epstein-Barr virus, EBV. Most cases, 80%, are due to mutations in the SLAM-associated protein, SAP, gene. SAP is involved in activating NK, T, and B cells. SAP defects impair control of EBV, often leading to fatal infectious mononucleosis, B-cell lymphomas, or hypogammaglobulinemia after EBV infection. SAP is also needed for T follicular helper cell function, impairing germinal center formation and high-affinity antibody production. Severe response to EBV is the main manifestation.
Finally, common variable immunodeficiency, CVID, main features.
CV-AID involves low IgG, IgA, and often IgM, with poor antibody responses to vaccines, leading to increased infections. Unlike XLA, it affects both sexes and usually presents later, childhood, adolescence, adulthood. It's heterogeneous, cause often unknown. Hallmark is recurrent sanopulmonary bacterial infections. Some get herpes virus infections, enteroviral meningitis, persistent giardia, diarrhea. Histology often shows hyperplastic germinal centers. B cells proliferate but fail to fully differentiate into plasma cells. Patients also have higher risk of autoimmunity, granulomatous disease, and lymphomas.
That was incredibly thorough on primary immunodeficiencies. Now let's move to the major acquired immunodeficiency. Acquired immunodeficiency syndrome, AIDS.
Right. AIDS is caused by the human immunodeficiency virus, HIV, a retrovirus, two main types, HIV-1, most common globally, and HIV-2, mainly West Africa, India. Transmission is via specific body fluids. Sexual contact, risk increased by other STDs, parenteral exposure to blood, 4B drug use, needle sticks, and mother-to-child, pregnancy, birth, breastfeeding. Blood transfusion risk is now virtually zero in developed countries due to screening.
What are the key parts of the HIV virus itself?
HIV is spherical. It has a dense, cone-shaped core capsid made of P24 protein. Inside are two RNA genomes and key enzymes. Reverse transcriptase, integrase, protease. Around the core is the matrix protein P17. The outermost layer is a lipid envelope taken from the host cell, studded with viral glycoproteins GP120 and GP41. These are crucial for infection. GP120 binds the cell receptor, CD4, and GP41 mediates fusion of the virus and cell membranes.
And how does HIV infect a cell and replicate step by step?
It starts by infecting cells expressing the CD4 receptor, mainly CD4 plus T cells, but also monocytes, macrophages, dendritic cells. Viral GP120 binds to CD4, but that's not enough. It then needs to bind a co-receptor, usually the chemokine receptor CCR5 or CXCR4. Different HIV strains prefer one or the other. R5 strains use CCR5, often infect macrophages early. X4 strains use CXCR4, infect T cells, often emerge later, more aggressive. HIV infects memory-activated T cells better than naive ones. Binding both CD4 and co-receptor changes GP120 GP41 shape, exposing a fusion peptide in GP41. This inserts into the host membrane, fusing the membranes, and the viral core enters the cytoplasm.
Then what happens inside?
Reverse transcriptase copies the viral RNA genome into double-stranded DNA, proviral DNA. This proviral DNA goes into the host nucleus. Integrase inserts it into the host cell's chromosome, becoming a permanent part. The integrated provirus is then transcribed by host machinery into new viral RNA and translated into viral proteins. These assemble at the cell membrane with viral RNA. New particles bed off, taking host membrane as their envelope. Finally, viral protease matures the new variants, making them infectious.
What's the core pathogenesis leading to the profound immunodeficiency in AIDS? What's the main target?
The main target is the immune system, especially CD4 plus T helper cells, but it also hits the CNS. The immunodeficiency comes primarily from the infection and death of CD4 plus T cells. Losing CD4 plus T cells devastates cell-mediated immunity and cripples humoral immunity, since T cells help B cells. CD4 plus cell loss happens several ways. Direct killing by the virus, replicating and budding. Apoptosis, programmed cell death, triggered by chronic immune activation in both infected and uninfected bystander cells. Reduced production from the thymus. Remaining CD4 plus cells might also be functionally impaired. HIV also infects and impairs macrophages and DCs, reducing their ability to fight infection and present antigens. Late stage sees destruction of lymph node architecture. HIV can even form syncytia, fused cells, which die quickly.
What's the typical course of untreated HIV infection from start to AIDS?
Usually 3 phases. First, the acute retroviral syndrome, weeks after infection. High viremia, virus spreads, often flu-like symptoms, temporary CD4 drop, then partial recovery. Second, the chronic phase, or clinical latency, can last years. Person feels okay, but HIV replicates continuously in lymphoid tissues, gradually destroying CD4 plus cells. Viral load is lower but steady. Third, clinical AIDS, defined by CD4 plus count below 200 cells L, or in AIDS defining opportunistic infection or cancer. Immune system is severely compromised, viral replication surges leading to opportunistic diseases. The immune system tries to fight HIV, CTLs, antibodies, but fails long-term due to CD4 destruction, HIV's high mutation rate, antigenic variation, and its ability to down-modulate MHC class I to evade CTLs. A switch to CXCR4 using virus can accelerate decline.
What are the hallmark clinical features of AIDS itself?
Diverse signs of immunodeficiency, common. Persistent fever, weight loss, diarrhea, swollen lymph nodes. But the hallmarks are opportunistic infections and certain cancers. Opportunistic infections cause most deaths in untreated AIDS. Examples: pneumocystis pneumonia, PCP, toxoplasmosis brain, cryptosporidiosis, diarrhea, candida esophagus, lungs, cryptococcus, meningitis, disseminated fungal infections, coccidioides, histoplasma, atypical mycobacteria, MAC, tuberculosis reactivation, CMV, retinitis, colitis, herpes simplex, varicella zoster. ART has drastically reduced these.
And the cancers.
Higher risk of Kaposi sarcoma, caused by HHV8, certain B-cell lymphomas, some linked to EBV, invasive cervical anal cancer linked to HTV. This is due to failure of immune surveillance against these oncogenic viruses. Neurologic issues are also common. HIV-associated dementia, opportunistic CNS infections, primary CNS lymphoma, wasting syndrome, and cardiac problems can also occur. The CDC classifies HIV based on clinical stage and CD4 count. 200 is AIDS immunologically.
Anna, let's cover amyloidosis, which you mentioned can relate to chronic inflammation. What's the fundamental issue?
Amyloidosis is about extracellular deposition of misfolded proteins that aggregate into insoluble fibrils called amyloid. Many different proteins can do this. Despite diverse precursors, all amyloid deposits look similar ultrastructurally: 7.5 10 millimeter non-branching fibrils. They have a characteristic beta-pleated sheet structure detected by X-ray diffraction. The diagnostic hallmark is binding Congo red dye and showing apple-green birefringence under polarized light.
What causes these proteins to misfold and deposit? How is it classified?
It usually results from either overproduction of aggregation-prone proteins, mutations causing unstable folding, or defective clearance of misfolded proteins. Classification is biochemical, based on the specific precursor protein, and clinical, systemic versus localized underlying cause.
What are the major forms? Precursor proteins.
Primary amyloidosis, AL, is most common systemic form. Deposits are AL protein from immunoglobulin light chains, lambda more often than kappa, made by monoclonal plasma cells, often underlying myeloma or MGUS. The free light chains are the precursor. Localized forms can also be AL. Reactive systemic amyloidosis, AA, involves AA protein derived from serum amyloid A, SAA, an acute phase protein. It's secondary to chronic inflammation like rheumatoid arthritis, IBD, chronic infections, some tumors. Hemodialysis-associated amyloidosis involves B2 microglobulin apoptosis deposits in long-term dialysis patients because kidneys can't clear it. Hereditary amyloidosis involves various mutated proteins like transthyretin, ATTR, and familial amyloidotic neuropathies, and systemic senile amyloidosis. Localized amyloidosis is confined to one site. Examples: nodular AL in lung larynx, endocrine amyloid in medullary thyroid cancer, a cal from calcitonin, or in type 2 diabetes islets, AIAPP from islet amyloid peptide.
Where do these deposits typically occur and what damage do they cause?
Organ pattern varies by type. AA often hits kidneys, liver, spleen. AL often involves heart, GI tract, nerves, skin, tongue. Grossly, organs may be enlarged, firm, waxy. Histologically, it's always extracellular, starting between cells near basement membranes, eventually compressing and destroying cells. Vascular deposits are common, especially in AL. Kidney involvement is common and serious. Deposits in glomeruli cause proteinuria, nephrotic syndrome, renal failure. Spleen gets enlarged, SIGO or lardaceous spleen. Liver gets enlarged. Amyloid deposits in space of DISC can cause atrophy, but function often preserved. Heart involvement, any type, especially AL and senile ATTR, causes restrictive cardiomyopathy, heart failure, conduction problems. GI tract, tongue enlargement, macroglossia, motility issues, malabsorption, bleeding.
What clinical features might suggest amyloidosis?
Often nonspecific early weakness, weight loss, lightheadedness. Later, more specific signs based on organ involvement. Nephrotic syndrome, restrictive cardiomyopathy, heart failure. hepatomegaly, macroglossia, peripheral neuropathy, easy bruising. Diagnosis requires tissue biopsy showing Congo red positive deposits with apple grain birefringence. Then you need tests to identify the specific amyloid protein type.
That concludes our incredibly in-depth exploration into the diseases of the immune system. We've truly gone deep today, covering an immense amount from the normal response right through hypersensitivity, autoimmunity, immunodeficiency, transplantation, and amyloidosis.
Yes, and as you can really appreciate now, understanding the normal immune system, all its cells, tissues, receptors, signals, is just absolutely essential for grasping how this huge spectrum of diseases arises when things go wrong.
It's also clear this is a dynamic field, lots of ongoing research, many aspects still being figured out. The immune system's balance is truly remarkable, and when it's disturbed, the health consequences are, well, profound.
Which leads us to a final, maybe thought-provoking question for you, our listener. Given how intricate and crucial the immune system is and how delicate its balance, what might be the long-term implications of our, increasingly sterile modern environment on the development and prevalence of immune-related diseases like allergies and autoimmunity? Something complex to consider as we keep learning about this vital system.